Welcome to the first slide on enzymes, and this slide looks at the roles of catalyzing enzymes in catalyzing reactions and the effect on metabolism, which is a word we'll look at and explore at a cellular and whole organism level. Okay, and also to include the idea that enzymes affect both structure and function. So we'll try and look at all of those things in turn. If I move down and look at the diagram here, try and illustrate the idea of metabolism. Here I have a cell, and the cell here has got metabolism, which are all the biochemical reactions that occur within this cell. Each of these biochemical reactions will be catalyzed by a specific enzyme. Okay, so there are maybe many thousands of biochemical reactions occurring surrounding the cytoplasm of this cell. Okay, and we call all of those reactions together metabolism. Each single reaction, biochemical reaction, will be catalyzed by an enzyme, and that enzyme will only specifically catalyze one particular reaction. Okay, within metabolism there are two basic types of metabolism that go on inside a cell. One is called catabolism, or catabolic reactions, and one is called anabolism, or anabolic reactions. Okay, catabolic reactions take large molecules and break them down into smaller molecules. Okay, respiration would be an example of a catabolic reaction, which takes things like glucose or fatty acids and breaks those quite large molecules down, strips the hydrogens off and reacts them with oxygen to produce water, and also strips the carbons off and reacts those with oxygen to produce carbon dioxide, and that releases the energy which is stored within the fatty acids or the glucose. So that would be an example of catabolism. Okay. Also, we have anabolic processes that occur inside cells. So we have examples, things like protein synthesis would be an example of an anabolic process where we take simple subunits such as amino acids and cobble them together to form large protein molecules, okay, such as a collagen molecule, which might be a structural molecule which is produced in bone or teeth or cartilage, etc. Okay, so that would be an anabolic process. Often anabolic processes require energy, okay, and that ATP is released from the catabolic breakdown of things in processes such as respiration. Okay, so that gives you an idea of what metabolism is all about and how enzymes are involved in metabolism. If I move back to the slide here, just to illustrate some of the other things, so we talked about the fact that enzymes can work at a cellular level. Okay, so we talk about lots of these enzymes working within cells, but also they're important at an organism level because some of these enzymes work outside of cells, such as the digestive enzymes we studied at GCSE. Things like amylase, which breaks down starch, is produced in cells but released outside of the cell and operates in the gut. Okay, so that'd be an example of how an enzyme might affect a whole organism. Another way of looking at the importance of enzymes is to see what happens when enzymes go wrong, or enzymes are faulty in some sort of way. And there are a number of inherited diseases which result in a faulty enzyme being produced. Okay, and I'm going to show you two examples of these. One is an example called Tay-Sachs disease. Okay, and Tay-Sachs disease is a mutation in a gene which is responsible for encoding for an enzyme called hexosaminidase, or Hex-A for short. Okay, and what this does is this enzyme is responsible for breaking down a substance which is absorbed by nerve cells called a ganglioside. Okay, now gangliosides enter nerve cells, okay, and in healthy individuals are broken down in the lysosomes of healthy nerve cells. For sufferers of Tay-Sachs disease who don't produce an effective hex A enzyme, they can't break down these gangliosides, and the result is the gangliosides accumulate in the cell bodies of their cells, and their nerve cells eventually die. And unfortunately for sufferers of this disease, the sufferers don't tend to live very long. In fact, they very rarely survive longer than 4 years old. Okay, so it's a devastating genetic disorder. Another, so if I show you very quickly show you that diagrammatically down here, and then I can show you a little animation of it as well. Okay, so here I have, in a healthy child, here is the cell body of a nerve cell, and we have these structures, membrane-bound packets full of lysosomes which contain enzymes which break things down. Now, in a healthy individual, these gangliosides enter into these lysosomes and then are broken down by this enzyme hex A. Okay, so that's not a problem. For a child with Tay-Sachs, what happens is that the ganglioside enters the lysosomes but isn't broken down because hex A is not effective at breaking that down, and therefore these lysosomes accumulate with this ganglioside within the cell body and eventually kill off the cell. If I can show you below an animation which will help you understand what's going on there.
Disease allele from both parents. The hex A gene encodes an enzyme called beta-hexosaminidase A, which is an enzyme necessary for the normal degradation of GM2 ganglioside. The Tay-Sachs mutations are severe and cause cells to make dysfunctional enzyme with no biochemical activity. In the developing brain of a young child, GM2 ganglioside is rapidly made and degraded. If the child doesn't have any beta-hexosaminidase A activity, because both copies of their hex A genes are producing dysfunctional enzymes, then GM2 ganglioside builds up in the neurons and eventually destroys them. Because they don't have any functional beta-hexosaminidase A enzyme in their neurons, individuals with Tay-Sachs disease cannot clear the excess GM2 ganglioside. Finding a treatment for Tay-Sachs disease has proven very difficult because nobody has yet found a good method of clearing excess molecules or replacing enzymes within cells. Hopefully one day, gene therapy or some other treatment will be able to address these problems and cure this disease. Children affected by Tay-Sachs disease appear normal for the first few months of their lives. However, as the GM2 ganglioside starts to build up in their neurons, the mental and physical abilities of these children deteriorate. As the disease progresses, the child becomes blind, deaf, and loses the ability to swallow. Dementia and seizures are also common. Eventually, the child is paralyzed before the disease finally kills them. It is a truly horrific disease, and unfortunately, there is currently no medical treatment that can stop or slow its progression. Let's review.
So moving on to discuss another example of one of these mutation diseases which results in a faulty enzyme is a disease called phenylketonuria, or PKU for short. Okay, this one affects an enzyme which is involved in breaking down an amino acid which is called phenylalanine. Okay, so as you remember, proteins are made of amino acids. There are 20 different types of amino acids that constitute proteins. One of these is called phenylalanine. Okay, so when you eat protein, what happens is that those proteins are broken down into amino acids in the gut, okay, by enzymes such as pepsin and trypsin. Okay, those enzymes, those amino acids are then absorbed through the gut wall into the bloodstream. Okay, they're then taken to the liver, and if those amino acids are required for protein synthesis, That's excellent. If they're not, they're broken down. In a healthy individual, the excess phenylalanine is broken down into harmless substances. If the enzyme doesn't work, as it doesn't with sufferers of PKU, what happens is the phenylalanine accumulates in cells and eventually in the bloodstream, and it has toxic effects on the development of nervous tissue. And again, I can show you a little animation below here of the effect of PKU.
Phenylketonuria, or PKU, is an inherited disease. It can cause mental retardation if not treated early and for life. PKU is rare. Only 1 out of every 15,000 babies in the United States is born with it. PKU is more common in people whose ancestors were Native American or Northern European. Babies are checked for PKU right when they are born. PKU is treated with a special diet. Without treatment, people with PKU become mentally retarded. This health education tutorial explains what PKU is. It discusses its causes, symptoms, diagnosis, and treatment. What is PKU? Our bodies need proteins to grow, make new cells, and repair tissues. Proteins are made of 20 building blocks called amino acids. Think of amino acids as 20 different Lego blocks. They go together to make different shapes. Protein comes from meat, fish, milk, eggs, beans, and nuts that we eat. The body breaks protein down into amino acids. Any amino acids that are not needed are broken down and disposed of in the urine. One amino acid is called phenylalanine. People with PKU cannot process phenylalanine. If phenylalanine is not processed, it builds up in the blood. This buildup affects nerve cells, causing brain damage and mental retardation. Symptoms. Babies born with PKU act like any other baby for the first few months. If the PKU is not treated, they start losing interest in things when they are 3 to 6 months old. When kids with untreated PKU are 1-year-old, it is obvious that they are not developing like they should. Children with untreated PKU have nerve and brain damage. Untreated PKU sometimes makes the child smell musty. This is because the buildup of phenylalanine is in their breath, urine, and sweat. They may also have dry skin, rashes, or convulsions. Kids with untreated PKU often have a small head called microcephaly. The body processes phenylalanine into tyrosine, which produces melanin. Melanin causes darker skin and eyes. The body of kids with PKU cannot process phenylalanine into tyrosine. That is why kids with untreated PKU usually have very light skin and blue eyes. Luckily, problems from PKU are rare. Most babies born with PKU are diagnosed and treated early. Causes.
So that completes a look at our first slide here, okay, with some key ideas to pick up, the words, keywords, catalysis, metabolism, catabolism, anabolism, okay, and then I've given you 2 examples of dramatic effects inside the functioning and structure of individuals if enzymes don't work: Tay-Sachs disease and phenylketonuria. The second slide in this first screencast on enzymes looks at distinguishing between 2 different types of enzyme, one which we call intracellular enzyme or intracellular enzymes, and what we call extracellular enzymes. Intracellular enzymes, which catalyze intracellular reactions, are produced in cells and have a site of action inside the cell, okay, or attached to the inside of the cell membranes. Okay, so an example of that would be an enzyme called catalase, which is an enzyme you'll come across in various practicals that you do, and catalase is responsible for breaking down hydrogen peroxide in cells. You may ask, why on earth would we get hydrogen peroxide in cells? And the answer is it's a waste product from the breakdown of a number of substances, including fatty acids and phospholipids in cells. And hydrogen peroxide, if it accumulated in the cell, it's a very strong oxidizing agent, and it'd be very poisonous. So it has to be very rapidly broken down, and that occurs inside specialized membrane-bound vesicles inside the cell, which are called peroxisomes. Okay, so inside these membrane-bound vesicles, we encounter the enzyme catalase, which breaks down the hydrogen peroxide to water and oxygen. Okay, so the catalase would be an example of an enzyme which has a site of action inside a cell, okay, and therefore that's an intracellular reaction, and we describe it as an intracellular enzyme. As opposed to extracellular enzymes which catalyse extracellular reactions, and these are reactions which occur outside of the cell. Now, extracellular enzymes are produced inside cells, and then they are secreted outside of the cell where they catalyse extracellular reactions. Okay, so two examples of that are two enzymes you came across at GCSE. One is called amylase, which hydrolyzes starch to maltose, breaks the glycosidic bonds between alpha glucose molecules, and breaks down this polysaccharide starch into this disaccharide maltose. And it is produced in cells that line the salivary glands, okay, around the mouth, and also produced in the pancreas, and is released into the small intestine from the pancreas. So it's produced in pancreatic cells and produced in salivary gland cells. The other example is trypsin. Trypsin is a protein digesting enzyme, and it is its site of action is in the small intestine. Okay, it's produced as an inactive form called trypsinogen, and it's produced inside cells of the pancreas. Okay, so this little diagram below should help to illustrate that. Here is a cell, and this could be a cell lining the salivary gland, okay, or it could be a cell in the pancreas lining the pancreatic duct. Okay, the principles are exactly the same. So as you'll notice, the cell has various organelles, including rough endoplasmic reticulum with ribosomes. Okay, so this is the site of a protein synthesis, a type of anabolic reaction that happens inside these cells, where amino acids are cobbled together into a specific structure with a specific sequence of amino acids, which then folds up into secondary structures and tertiary structures to form an enzyme in this particular case. And it could be amylase if we're dealing with a cell that produces this particular enzyme, or it could be a cell that produces trypsinogen, which is the inactive form of trypsin. Okay, so what happens is that enzyme is produced inside the cell, okay, and it then is exported to a structure called the Golgi body, where it is modified and packaged, and then vesicles take it from the Golgi body, secretory vesicles take it to the cell surface membrane, where it's released into the outside world through a process called exocytosis. Okay, and then that releases it into some sort of duct, which then takes it to its site of action. Okay, for amylase, that could be in the mouth. Okay, if it's produced by a salivary gland, or it could be, if it's produced by the pancreas, the site of action could be in the small intestine. For trypsin, it's produced in the pancreas by pancreatic cells, released into the pancreatic duct in its inactive form called trypsinogen. Okay, and then it's transferred to the small intestine, where it's then converted into its active form trypsin, which then can break down the peptide bonds that hold proteins together, thus digesting the protein. So that concludes the second slide. So just to summarise, you need to know about the word intracellular and what that extracellular reactions, what they are. and you need to know examples. Catalase, okay, being an example of intracellular reaction, and you need to know two examples of extracellular reactions, one catalyzed by amylase and one catalyzed by trypsin. This third slide of the first screencast on enzymes looks at a number of facets of enzyme activity. First of all, how does an enzyme affect catalysis of a biochemical reaction? The answer to that is it does that by lowering the activation energy, or creating an alternative pathway for the reaction which has a lower activation energy. What does that actually mean? If you look at this graph over here, I'll blow that up for a second. If you look at this graph over here, we have energy in substrates and energy in products. This is what we call an exothermic reaction, i.e. there is more energy here in the substrates than we have in the products, and therefore as the reaction goes, this amount of energy here would be given out, okay, as an exothermic reaction. Now, for any reaction to occur, we first of all have to initiate the reaction by putting some energy into the system, usually to break bonds, initial breaking of bonds, and that is called activation energy. Okay, now this results in lots of collisions between molecules being unsuccessful because the activation energy isn't supplied, and therefore the reaction doesn't happen. If we are able to provide an alternative pathway for the reaction, which has got a lower activation energy, there is more likelihood of collisions occurring being successful, and therefore more product will be formed, and therefore we'll get a greater rate of reaction. Okay, so that's how enzymes perform catalysis, is that they reduce the activation energy. In actual fact, it's exactly the same way as any catalyst manages to speed up the rate of a biochemical reaction. Okay, so that's the first thing, and if I look down here, there's a little animation to try to illustrate that.
For any reaction to occur, even one that releases energy, some energy must be added to get the reaction going. This energy is needed to break bonds in the reactant molecules. The energy needed to start a chemical reaction is called the energy of activation, EA. This required energy input represents a barrier that prevents even energy-releasing reactions from occurring without some added energy. How does a living cell overcome the energy barrier so that its metabolic reactions can occur quickly and precisely? A special kind of protein called an enzyme is the answer. An enzyme serves as a biological catalyst, increasing the rate of a reaction without being changed into a different molecule. An enzyme does not add energy to a reaction. Instead, it speeds up a reaction by lowering the energy barrier. An enzyme is very selective. Its three-dimensional shape allows it to act only on specific molecules, referred to as the enzyme's substrates. As the substrates bind to the enzyme's active site, they are held in a position that facilitates the reaction. This takes less activation energy than the unaided reaction. Products form and are released. The enzyme emerges unchanged from the reaction. Because of the specific fit between enzyme and substrate, each enzyme can catalyze only one kind of reaction involving specific substrates. Thousands of different enzymes may be required to carry out all of a cell's metabolic processes.
Now, if I move to the next bit of this slide, this looks at a number of models or hypotheses as to how an enzyme is able to lower the activation energy. The first of these manages to explain the specificity of an enzyme. What do I mean by specificity? We find that enzymes can only be responsible for one particular substrate, converting one substrate into one particular product. The reason for this is that an enzyme has a tertiary structure, a specific tertiary structure. Okay, so if you go back to protein structure, we have a specific sequence of amino acids, and the sequence of R groups in that primary structure of amino acids causes a particular type of folding, which we call secondary folding, and then the domains of secondary structure then fold in on each other to form this tertiary structure, which has a specific three-dimensional shape. Now, part of the surface of that three-dimensional shape has a region which we call the active site, and that active site has a specific shape and a specific chemical structure, and that is complementary in shape to the substrate that that enzyme is going to bind to. The consequence of that is any other substrate will not be able to bind to that active site because it is not complementary in shape to that active site. Okay, so hence enzymes can only be responsible for catalyzing a reaction involving one particular type of substrate. Okay, now that is, we have a hypothesis which tries to explain that idea of specificity, which is called the lock and key hypothesis, which very simply looks at the idea of a lock and a key. So we have a key here, which is analogous to the substrate, and we have the lock here, which is analogous to the enzyme, and the key can fit into that enzyme. Okay, so only this specific key can fit into this lock, i.e. only the specific substrate shape can fit into the complementary shape of the active site and the enzyme. Okay, so that's shown over here, substrate fitting specifically into this active site because this is complementary. Be careful not to say that it's the same, because the shape is not the same, it is complementary to the shape. Okay, so that's the lock and key hypothesis. And the lock and key hypothesis is a very neat way of explaining the specificity of enzyme. What it doesn't do, however, it doesn't go any way to explain how this notion of catalysis works. How does the fitting of that key into that lock or that substrate into that enzyme, how does that lower the activation energy? So there's a more recent hypothesis which is known as the induced fit hypothesis, and what that does is it tries to explain the specificity of enzymes, but also at the same time tries to explain how the catalysis occurs. So the idea of the induced fit hypothesis is that the enzyme doesn't fit the substrate exactly, in that when the substrate comes together with the active site to form what we call an enzyme substrate complex, the enzyme has to mould around the substrate. And in so doing, what happens is that there's a redistribution of charge and electrons around the substrate, which in some way makes the reaction to form the product more energetically viable, okay, thus reducing the activation energy. Okay, so that's the idea of the induced fit hypothesis. And if I look below, I've got some animations to show you of these various things. So if I look at this first one here, which actually shows a little bit about both of these lock and key hypothesis and induced fit hypothesis.
Enzymes are proteins which catalyse metabolic reactions involving the conversion of a substrate into a product. Enzymes bind with substrates due to their complementary shape. This is called the lock and key model. Rather like a spaceship docking into a space station. In the induced fit model, the enzyme changes shape to accommodate the substrate. Enzymes enable metabolic processes to proceed rapidly at room temperature by providing an alternative reaction pathway of lower activation energy. Without enzymes, these reactions would happen too slowly to sustain life, or would require extreme conditions of pH or temperature. Carbonic anhydrase can...
And then below there's yet another little animation which I'll talk you through here. So here we have the enzyme which is coming in and binding to our active site. The enzyme then molds around the substrate, causing redistribution of charge and electrons, which then makes gives an alternative pathway. Then the product is formed, which then dissociates from the active site, and then the enzyme returns to its original shape and is available to bind to a further substrate molecule. So just making sure that we've gone through all these terms, these key terms that you should be aware of. Tertiary structure, that goes back to our study of proteins, okay, the tertiary structure of proteins. Remember, enzymes are proteins, they have specific tertiary structures. On that tertiary structure we have a specific region called the active site, which binds to the substrate, and they have to be complementary in shape to each other, which explains the specificity of enzymes, i.e. one enzyme will only bind to one particular type of substrate molecule, and that's known as its specificity. You should understand what we mean by the lock and key hypothesis. You should understand that when an enzyme binds to a substrate, a chemical association occurs between these two substances, which we call an enzyme substrate complex. That results in an alternative pathway for the reaction to occur, which reduces its activation energy, which results in the production of an intermediate called the enzyme product complex. And the enzyme, because the product isn't specific in shape to the enzyme anymore, what happens is the product then dissociates, is released from the active site to produce the product. Okay, so you should understand what all those terms mean. And that concludes our first screencast on enzymes.